Alright, now let's move on to specification number 4. This one asks us to explain how enzymes are specific, basically, how their active sites have a complementary shape that fits exactly with their substrate. It's often described using the lock and key model, where the enzyme is the lock and the substrate is the key. Now, when we talk about enzyme specificity, we're really referring to how particular enzymes are about the substances they work with. First of all, enzymes are highly specific, they only act on certain substrates, and that's because of the unique shapes. The active site of an enzyme, which is where the substrate binds, has a shape that's exactly complementary to the substrate. This idea is described by the lock and key hypothesis, where the enzyme is the lock and the substrate is the key. This specific shape comes from the way the enzyme's chain of amino acids folds into a unique three-dimensional structure. Because of that, only substrates with the right shape can fit in and bind properly. Also, it's worth mentioning that many biological reactions don't just happen in one go, they often occur in several steps. And for each step, a different enzyme is needed. Not only are enzymes specific to a substrate, but they're also specific to the type of reaction. For example, a hydrolase enzyme will only catalyze a hydrolysis reaction, it won't work on an oxidation reaction. Now, when a substrate binds to its enzyme, they form what we call an enzyme-substrate complex. This allows the reaction to happen very efficiently. To give you an idea of how fast this can be, catalase, an enzyme found in many cells, can break down about 40,000 molecules of hydrogen peroxide every second. Without the enzyme, the reaction would be way too slow. And finally, enzymes can be specific to the type of bond they break, or the type of molecule they build. Take amylase, for example, it's an enzyme found in saliva that breaks down starch into maltose, a sugar that's much easier to digest. As you can see here, we've got different locks with their own unique keyholes, and each one has a specific key that fits it. No two keys can open the same lock unless they're an exact match. Now, this is a great way to visualize what we call the lock and key hypothesis when it comes to enzymes. In this analogy, the enzyme is like the lock, and the substrate, that's the molecule the enzyme works on, is the key. Each enzyme has a specifically shaped active site, just like a lock has a uniquely shaped keyhole. And just like only one particular key can open a lock, only the right substrate can fit into an enzyme's active site and trigger a reaction. This explains why enzymes are so specific, they're built to work with one particular substrate, and nothing else will fit quite right. So, let's go through the lock and key model step by step. First, we start with two separate pieces, the enzyme and the substrate. The enzyme has a special region called the active site, and the substrate is the molecule that needs to be changed. Next, the substrate binds to the enzyme's active site, like a key fitting into a lock. This forms what we call the enzyme-substrate complex. Once they're connected, the enzyme does its job, it acts on the substrate and helps turn it into something new. At this stage, we now have an enzyme-product complex. Finally, the product is released, and the enzyme remains unchanged. Its active site is still intact and ready to go again with another substrate. And it's important to note, enzymes and substrates don't have the same shape. Instead, they have complementary shapes, meaning they fit together perfectly, even though they're different, just like a lock and its matching key. Let's take a look at this diagram. It shows a cycle that's a great example of how enzyme specificity works, especially in the case of building cellulose. We start with individual glucose molecules, which are the building blocks. Now in step 1, we see an enzyme, highlighted in purple, picking up a glucose molecule. This is important because enzymes aren't just picking up any random molecule, they're very specific. This particular enzyme only works with glucose, not with other sugars like fructose or galactose. Then in step 2, the enzyme brings the glucose to the start of a cellulose chain. That means the enzyme doesn't just grab glucose, it knows exactly where to place it, connecting it to an existing cellulose molecule. In step 3, the enzyme helps join the new glucose to the chain, forming the special chemical bond needed to make cellulose. This is where its catalytic power comes in. It doesn't just stick glucose anywhere, it forms a specific type of bond called a beta-1, 4-glycosidic bond, which is unique to cellulose. Next, in step 4, the cellulose chain grows longer as more glucose molecules are added one by one. And finally, in step 5, the enzyme lets go and is ready to repeat the process again. This shows another key feature of enzymes, they aren't used up in the reaction. They can keep going, again and again. So, what does this show us about enzyme specificity? First, the enzyme is substrate specific, it only binds to glucose, not other sugars. That's like the lock and key idea, only the right key, glucose, fits the lock, enzyme. Second, it shows reaction specificity, this enzyme isn't just sticking glucose onto anything. It specifically builds cellulose, not starch or any other molecule. And finally, we see product specificity, the enzyme's action always results in the same product, cellulose. So, in short, this diagram is a great way to see how enzymes are incredibly selective, They know exactly what to work with, what to do, and what to make. That's enzyme specificity in action. And that wraps up everything for Unit 5.1 on enzyme action. I've included the related notes in the comment section and also pinned them below for easy access. I hope you found the video helpful. If you have any questions, feel free to drop them in the comments, I'll be happy to help. Stay tuned for the next unit coming up in this chapter. Thanks for watching.